My name was already mentioned, that's also my background. So today I want to talk about the impact of intestinal cancer-causing genes on the immune system. Starting with very simple clues. Also cancer was perceived very simple in the beginning. So
wait.
is it not on? I think I switched it on.
Let's check. It doesn't go away from the time. Don't worry. It's on. Yeah, it should be on. Maybe. Yeah. It should be working. Maybe we can.
Test?
Maybe it's just behind your. Okay. You can start for it. Continue.
Is this now working?
Yeah.
Okay.
It's better.
Everyone else now? Okay.
Just speak a bit louder. No, sorry. And
Okay, let's make it very, very strange. I'll just hold it in front of my mouse.
you want to use this one?
Yes!
Yes? All right, problem solved.
Great. Should be.
Yeah.
Okay, now I can start a rap career with the microphone. Yeah. So we have like a very simple view of the cancer. So we have like a cancer cell, and we have like a cancer cell because it has like cancer genes which are activated. And these genes are leading that the cancer cells becomes more, it's proliferating. It becomes 2 cells, 4 cells, 8 cells, 16 cells until the patient dies. However, like as finally you get all the money, if you're related. However, nowadays cancer is like considered to be a little bit more complicated. So we have like lots of cells in the surrounding, and the cancer cells are not only proliferating but also communicating with these cells. And this communication is also influenced again by cancer genes. So this is something highly interesting, and we want to study it by using mouse models. So this is a mouse; it's not a kangaroo. And basically, these mice are genetically altered, so they have like new DNA. And what's new is that they have like a William promoter and a Cre recombinase. And before I talk loud, let me explain it to you. So the William promoter is simply a DNA sequence where only in the intestine certain proteins will bind, and therefore enable only the intestine that the polymerase will bind, which is like a reader for the DNA. Therefore, the polymerase can read the DNA code for the Cre recombinase, enabling it to become protein. And the Cre recombinase is a protein which is like a molecular scissor, again recognizing DNA elements. And therefore, this Cre recombinase will only cut out the intestine because only there it is like given. For example, a stop signal in front of an oncogene, a gene which upon activation can cause this cancer. Or reverse, it can cut out a tumor suppressor gene, a gene which normally protects from cancer. So these mice will all get tumors. Yes. And we want to study them. And for example, it was shown that tumor cells can secrete a protein called CCL4. And what CCL4 does, it attracts immune cells, and the immune cells can then attack the cancer cells. But for example, oncogene was shown basically blocks the secretion of CCL4. And to find out if maybe less or more immune cells are given in our models, we want to simply cut our frozen tumors, so we will have a very thin tissue layer. We put it onto a glass slide, and we throw antibodies on them. So antibodies are highly specific. They will label just very specific immune subtypes, and every antibody will be coupled to a fluorophore, which we can activate by laser, not like the star lasers constantly used, but a very simple laser from a microscope. And then we will have a light signal, and we will see on this spot, there's this antibody, and therefore this type of immune cell. The next step is that every cell has proteins, and every cell will present cleaved proteins or cleaved peptides on its surface, which enables the immune system to recognize them and to kill the cell. But the cell can also express inhibitory proteins. For example, PD-L1, therefore the T cell will not attack it anymore. And we want to look if the mRNA is increased in these tumors. So the mRNA is between the DNA and the protein level, and maybe there will be more in certain tumor types. Therefore, also like maybe a protectional mechanism against the immune system. So therefore, is this okay? I'm just irritated by this time. And the last step is that we want to also see if the immune cells really have an effect. And this we can only see if we implant or transplant cancer cells into immune deficient mice and see if they maybe will have a worse survival than simply normal mice. And what we finally want to do is that we, of course, want to find out new mechanism how really a cancer cell is interacting with its environment, with the cell surrounding it. And therefore maybe there are new possibilities also for treatment. So the patients will die later, so you will also get your money later. That's another topic. And finally happen basically these guys, so the medics. I think I'm ill in time, but I was completely irritated with this time. But that was it already.
You get extra time, don't worry. It was technical issues, don't worry. Thank you, Tim, and thanks for not being irritated and stressed by technical issues.
I think so.